Eccoci,
everyone.
buonasera a tutti. Allora,
The purpose of this
lo scopo di fare
roundtable
questa
is
tavola rotonda è
to
un po' di fare
draw
somme
some
di quello che è
conclusions
stata
based
la
on what we
giornata di
heard
oggi
today.
e
And the
inoltre da parte
association,
dell'associazione
insieme,
insieme con la
and the US counterpart,
americana della
the
associazione
Acute
Acute,
Syndrome Association,
ci sarebbe la
the idea
proposta
is
di
to
un
have a grant be made
c'è
available for a
anche
research
nelle vostre
project.
cartelline
You should have
trovate un
in
questionario
your dossier a
dimmi pure Alba.
questionnaire
Trovate un
which was
questionario
prepared
che
by
è stato
both
predisposto dalle due
associations. It
associazioni
would be a good
e che
idea to
sarebbe
fill
utile
it in
compilare
because it
perché
might give
potrebbe dare delle indicazioni per facilitare le
information
associazioni anche a
to the
prendere
associations
determinate decisioni di
so that
come
they can
investire
decide
il
how
loro
to
denaro nella
invest
ricerca.
their money in research. So please
Quindi è importante
fill in the
compilarlo e
forms and
restituirlo
return them to the
agli organizzatori.
organizers.
Per quanto riguarda
Now, on to our
la tavola
roundtable.
rotonda, volevo fare
I'd
un
like to very briefly
brevissimo summary di
summarize the
quelle che
main
sono
concepts that
stati
were
i concetti
expressed
espressi
during
durante
the
la
day,
giornata
and
e in
then
seguito
possibly
eventualmente
invite
invitare i
the
relatori della
speakers
giornata
to sit
qui a sedersi
around
tutti
the
intorno
table
e
with me
iniziare
and
un
start a discussion
dibattito e discussione
on the
sui
information that
punti
was
che ci sono
discussed
stati
this
oggi.
morning.
Ehm,
Just
Voglio fare
some
pochi
brief
flash su quelle che sono state
highlights
le presentazioni della
concerning the
giornata.
presentations, starting with
Partendo dalla presentazione di Riccardo Dolmec di questa mattina
Ricardo's
sulle
presentation
IPS,
this morning on
la
IP.
considerazione che credo
SCs.
di
The
sia
interesting
interessante
remark
è
is
che
that
sebbene la metodica
although this
potrà avere
method
numerosi
may
sviluppi
lead
per
us to
comprendere il
better
meccanismo
understand the
con cui
mechanism
appunto si determina la malattia da parte della proteina
responsible for
alterata,
illness
non
and
sembra
disease,
sia
it
chiaro
doesn't seem
qual è
clear
al
what
momento la popolazione
neurons
neuronale
can be
sul
used
quale
to study
studiare
the
il fenotipo.
phenotype. Although
Sebbene rispetto alla presentazione di Maria Passafaro
there is one
un'indicazione
indication,
viene
based on what we heard
dalla
from Maria
una delle
Passavaro,
ipotesi potrebbe essere quella
one
di studiare dei neuroni
possibility might
GABAergici,
be that of
ma
studying
questo sembra che
GABA
vada
neurons, but that needs
confermato.
to
Una
be
cosa ancora
confirmed.
interessante
Another
che non è
interesting
sempre la presentazione della
point that
Passafaro,
emerged
ma
from
anche è stata
Passafaro's presentation,
citata
but
da
which was also mentioned by
Getz,
Professor Getz,
è
is the
l'ipotesi che di un
hypothesis that there may
effetto
be an
della
effect of the
proteina
protein on
sulla
neuromigration.
migrazione neuronale. Abbiamo visto
We saw
delle
pictures
immagini di
of
un
the
ippocampo con
hippocampus
una
with
normale
normal
rappresentazione degli strati
neuron
e
layers.
questo, diciamo,
But
tornando
going
indietro
back
all'IPS
to
e rifacendoci
IP
all'ipotesi della migrazione,
SCs and the
potrebbe essere
migration
utile
hypothesis, it might
effettuare
be a
uno
good idea
studio
to
nella fase
conduct a study in
precoce
the
della
early phase of
differenziazione proprio per
differentiation
capire
to
se
understand whether or not there
vi è
is
o
a
meno un
migration
difetto di migrazione,
defect,
questo
and
sulla
that's what I can say
differenziazione
about
delle IPS.
differentiation of IPSs. Then
Dopodiché gli
the
altri
other
concetti
important
importanti
concepts
che
that I
diciamo
think we
vanno
need to
sicuramente
further
implementati,
stress
vanno
and
stressati
elaborate
nella
on in
rotonda
the
e anche
roundtable
utilizzati per dei possibili
are
progetti sono appunto
the
l'ipotesi,
neurosteroid
l'idea
hypothesis
formulata da
formulated by
Getz
Professor
che
Getz,
va,
which we
diciamo, che
are
noi stiamo
trying
cercando un po' di, come dire,
to support
osservare con dei
with
dati
clinical
clinici, ma
findings.
soprattutto cercare di capire se poi
What we want to find out
dall'animale si può
is whether the concept
trasdurre il
may be
concetto,
transposed
quindi
from animal
cercheremo
to
di analizzare,
human
di
subjects.
dosare
We're
questi
going to try
neurosteroidi
to see
nel
whether we can
sangue
use these
delle nostre
steroids in the blood
pazienti
of our patients
proprio
in order
per
to
accoppiare i
couple
dati,
in
diciamo,
vivo bioclinical
bioclinici in
data with
vivo
the
con i
data coming
dati
from the
che
lab.
vengono dal laboratorio.
Last but not
Non
least,
ultimo
we
poi ci
have all
sono
the
tutti i
clinical
concetti
concepts
clinici che
that will
verranno
definitely be
sicuramente
further developed
ampliati
in the
nella
last
ultima
part of our
parte della
day,
giornata, ma già
but I think
qualcosa si
that
può
a
anticipare,
few
già
have
qualcosa
already
è stato
been
anticipato.
anticipated.
Allora, prima di
First
tutto,
of all,
nonostante
although
sono
through
diversi
several
anni
years we've
che
been
ci si
studying
occupa di
this
questa
disease,
malattia,
it would
sembra
seem
che
that the
ancora la caratterizzazione
characterization
delle
of
crisi, la semiologia delle crisi non
seizures
sia
is not so
così
clear.
definita e
And this
chiara.
is
Questo
because
perché? Perché è importante la caratterizzazione delle crisi,
it's
così come
important
la
not to
caratterizzazione
only
degli sviluppi
characterize seizures,
cognitivi,
but also
perché
cognitive or intellectual disabilities,
partendo
because
dalla
based
precisa
on the
caratterizzazione
characterization
di crisi
of
cognitivi,
intellectual
soprattutto nel
disability and
follow-up, è
seizures,
possibile
it's
ipotizzare
possible
delle
to
soluzioni terapeutiche
hypothesize
differenti,
different
delle soluzioni
therapeutic
terapeutiche che
solutions,
possano
which can be
essere
more
più
or
o meno
less
efficaci sia
efficacious
riguardo
in
le
the
crisi,
case
ma
of seizures,
soprattutto
but
per disturbo
especially
del
in the
comportamento
case
ove
of
presente, dei tratti autistici ove
behavioral
presenti, che talora
disorders
sono
and autistic
quasi più
traits, which sometimes
invalidanti in
are
alcuni
even more
casi rispetto che le
invalidating
crisi stesse.
than the seizures
Ehm,
themselves. Then
E poi ancora
another
c'è da
aspect
ben
that is
definire
yet to be
la
clearly
differenza
defined
tra, sempre
is
dal
the
punto di vista
clinical
clinico,
difference between patients
tra i
with the
pazienti con
Dravet
sindrome di Dravet e
syndrome
i pazienti
and
con mutazione
patients with
protocaderina, che
mutation of
questa differenza
PCDH19.
diciamo
This is
in
a
varie
difference
riunioni
that's come up
italiane, ne
in
abbiamo
many
fatto
meetings
occasione di
in
incontri che abbiamo
Florence
avuto a
and in
Firenze, ma
other
anche in altri
meetings here in
meeting,
Italy.
sembra che
For us
per noi sia
now, the
abbastanza
idea,
chiara. Cioè,
well,
noi
the difference is
abbiamo
rather clear
definito
because
tra
we've
di
defined a
noi un
common
linguaggio
language
comune
amongst
e
ourselves,
quindi
and we don't have many
non abbiamo
doubts as
molti
to
dubbi
the
sull'overlap.
overlap. In other words,
Non
we don't
ipotizziamo un
think that
overlap
there
tra le
might be an
due
overlap
condizioni,
between the
però
two conditions, but
questo
that's
non
only
è
what we think.
evidentemente
This
così
is not
universalmente
universally
accettato, così
accepted
diffuso
as a
questo concetto,
concept.
tant'è che sia
Indeed, in the last
nelle
two
ultime due presentazioni
presentations, the
si è
concept of the
riproposto il concetto
overlapping
dell'overlap.
has come back to the fore.
Quindi in
So
base a tutti
based on
questi
all
diciamo,
of these
messaggi io
messages, I would like to
aprirei la
open up our
discussione e
roundtable discussion.
inviterei i
I'd like to
genetisti a
invite the
partecipare al tavolo,
geneticists to
quindi Riccardo Dolmec, la
participate in the
Passafaro,
roundtable,
il
Dr.
professor
Terracciano,
Terracciano,
Dr.
se vogliono
Dolmec,
venire
and
qui al
everyone else.
tavolo. Ok, iniziamo con
So let's begin
la presentazione di
with
Dolmec sulle IPS.
Ricardo's presentation
Allora,
on
rispetto ad una possibile
iPSCs.
popolazione di
Now,
un gruppo di cellule indifferenziate,
with respect to an
quale
undifferentiated group of
secondo
cells,
voi potrebbe
which do
essere
you
la
think
popolazione neuronale
might
più
be
adatta
the
a
most
studiare il
suitable
fenomeno? Cioè, in
neuron
quale tipo di
population, which
neuroni
might be
dobbiamo
best?
differenziare queste
In which
IPS
types
per
of
avere
neurons
dei
should we
risultati più affidabili?
differentiate these iPSCs to have more reliable results?
Yeah, so it's an excellent question. It's one that comes up all the time, right? So which kinds of cells should you make and which ones are the relevant for modeling a disease? And I think the best answer to that actually comes from expression studies. So you have to look and see which cells actually express the gene, and that gives you information about what kinds of cells you should make. So in the case of protocoherin 19, the thing to do is to make cortical neurons and hippocampal neurons because that's where it's expressed. And in fact, we know that our cells express protocoherin 19. So I think in this particular case, it's a relatively easy question because those happen to be kinds of neurons we can make. Did that make sense?
Sorry. Yeah. Aspetta Carla, prendi il microfono.
What about what about interneurons? I
Yes.
mean, there are a few pathologies like IRX mutations related to the pathology of interneurons.
Well, Dravet, right,
Yeah,
is
Dravet. Yeah,
related to the
really
pathology of
interesting
interneurons.
cells.
Yeah, so we've developed, I mean, not just us, but several groups have developed ways of making interneurons. And I would say that we are maybe 3/4 of the way there. We're quite good at making, so the scientific question is, can you actually generate precursors that come, so interneurons are generated from the mid and lateral ganglionic eminences, not from the ventricular zone. And so you can actually pattern cells to express the genes there. This is a gene called NKX2.1, which is sort of pathognomonic for that population. So we're pretty good at making that population of precursors. We can then differentiate them into interneurons. The only class of interneurons we're not very good at making are parvalbumin cells. And that's because parvalbumin cells are born late. And unfortunately, that's one population of cells that is implicated in some classes of disorder. So we're still working at that. But I think we can make inhibitory interneurons reasonably well. And that's the other kind of cells one would want to make.
Is it actually possible to do, I mean, would it be possible or a good idea or maybe not, to do nasal biopsy in patients and get some cells deriving from neurons directly from PCDH19 patients, going to the nasal, what do you think?
Yeah, it's a nasal epithelium. I don't think that there was a time when that might be the only way you would have of getting neurons. I don't think that there are many advantages to doing that anymore, because I don't think that those cells are really any better. I mean, they're olfactory neurons, and they're olfactory neurons that actually are really strange in the sense that they die and then they get replaced by new ones. So I think that you're better off just making neurons from blood or skin.
Just because they are actually cells deriving directly from patients, you would not have to reprogram these cells.
You know,
They might express PCDH19.
They might. I would say that when we first started doing this, there was some question as to whether there would be changes in the phenotype associated with the reprogramming. And I would say that we have more or less settled that question. So, I mean, not just us, but the field. I mean, it seems that when you reprogram cells, you do slowly accumulate mutations, but you accumulate mutations very slowly. And so, and in fact, we can see phenotypes associated with the cells. So I would say that given that that's pretty invasive and that those cells are not easy to culture, I think you're better off just getting blood samples, isolating CD34 positive cells, and reprogramming those. And I think the place where we need to improve things is in making neurons from stem cells, but now there are methods for doing this more quickly by introducing certain genes. So I think if it were me and if I was going to study this more broadly and I wanted to do this for a lot of patients and maybe use it as a way of trying to understand variability, I would not take samples from the nose. I would reprogram cells.
Okay, thank you.
Invece,
Did you compare the whole exomes?
Microphone. Okay.
Thank you. Do you compare the whole exome sequencing of derived neuron from fibroblast? And did you find any variants, de novo variants?
Yeah. So yes. So we've done that. So one of the things that you worry about when you make iPS cells or you're studying patient biopsies and you're manipulating them in the lab is that during the process of reprogramming or differentiation, you accumulate mutations. So this is more or less what we and other people have found. So you gain about one copy number variant, so one microdeletion or one microduplication about every 4 cell divisions. This, in fact, is the same as is in fibroblasts or in blood cells. There's some aspect of just cell division that generates some errors. It's a little bit better, actually, than it is in lymphoblast lines, where people actually do genetics a lot. There, you actually get about one copy number variant every 2 cell divisions. So there are definitely some de novo changes. And so the way we try to deal with that is we actually have a lot of different, first of all, we don't subclone the lines, right? So we just simply, so you're actually looking at deletions, but there are different deletions in different cells as a function of time, and we hope that that averages out. And in fact, that's probably what happens in the brain too. The other thing we do is we have multiple lines from multiple patients, and then we look for common phenotypes. So that's the approach. It's not perfect, but it's, you know, it's our solution, yeah.
e invece che ne pensi rispetto all'ipotesi di una alterata migrazione neuronale, così come abbiamo visto nelle presentazioni della dottoressa Passafare di Getz, di studiare le iPS prima di differenziarle? Cioè è importante capire anche a livello di cellule staminali che cosa succede, e a livello, per esempio, che cosa ne pensi, se è utile studiare le rosette neurali, cioè la prima struttura polare che si stabilisce nelle iPS.
Ma secondo me stavo pensando la stessa cosa.
I was thinking about the same thing. One interesting thing might be to silence iPSC cells that are normal, harvested from wild type, sorry. You can silence them, take out the protocaterin, and see whether these cells are differentiated, because if there's a programming problem, I don't know, I'm not an expert, but if you abate, if you take out protocaterin, you should have problems, I think. You have difficulties. The other question I was asking myself is what would happen if in iPSC cells we were to do away with protocaterin? What would happen to the GABA levels in the alpha-1 subunits too? I think that having available iPSC cells from patients, we can also study why we have this different phenotype. But I was wondering whether, Riccardo, you've tried to do an experiment of this type to abate, to eliminate a protocaterin and see whether the neurons differentiate when you take out the protocaterin.
We have tried to reduce protocadherin 19 levels and looked at some very basic aspects of differentiation as well as some other aspects of the cell biology of cells. I would say the results are not striking, but to some extent that depends on exactly how you do the differentiation assay. So we have this nice way of looking at differentiation by looking at gene expression in single cells and using this microfluidic technique. And we haven't analyzed that data yet, so we don't actually know if there are differences in differentiation. I would say that, the one thing I would encourage everybody here to do is when you are trying to identify a phenotype associated with the mutation, and this is something that I encourage my students as well as just other people who work on disease, is to try and take as unbiased an approach as possible. We'll try and look at as many different processes, because one of the problems that we have sort of as basic biologists is that we settle on a hypothesis very quickly. If you settle on a hypothesis very quickly, you prove the hypothesis. And that turns out not to be always the best thing. So in the ideal world, you have an assay for migration, an assay for gene expression, an assay for synapse formation, and you have a bunch of different assays so you can, in an as unbiased a way as possible, try and identify the consequence of the mutation. I was going to say, with regards to the migration phenotype, there are well-established epilepsies that are due to migration phenotypes, but they're almost always associated with heterotopias. And I was wondering if somebody could tell me if there are heterotopias associated with protocadherin mutations, maybe somebody who's done MRI or something. No. So, okay, well, how about no lysencephaly or micro or pachygyria or anything like that, right? Because many of the migration mutations seem to lead to these structural changes. So, it might be migration is just one population of cells that are difficult to see by MRI, right?
Ok.
Posso dire un'altra cosa?
Certo.
Qual è la tua esperienza invece
What is your
rispetto
experience with
alla riprogrammazione di
the
cellule in
reprogramming
modelli
of cells
X-linked?
in X-linked
In
models?
letteratura,
In the literature,
insomma, è riportato che
it's been
modellizzare,
reported
cioè
that
riprogrammare cellule di
reprogramming
maschi è più facile che riprogrammare
male
cellule
cells
di
is
femmine
easier than
per
reprogramming female cells
l'inattivazione del cromosoma
for
X.
the inactivation of the X
E
chromosome.
se secondo te
And
è
do you think that it's
importante quando si sceglie
whenever
il
you
paziente da
choose the
riprogrammare,
patient in whom to
stabilire
do
appunto
reprogramming, is it
l'allele su
important
cui
to
segrega la
establish
mutazione?
the
Se
allele
segrega
on which
sull'allele paterno o
the
sull'allele materno?
mutation is segregated, whether it's the maternal or paternal allele?
Yeah, so this is actually an issue, and one of the things that, and this is, I would say, not true for every X-linked mutation. So, for example, for Rett syndrome, people have managed to make iPS cells, and they make some cells. So just to sort of put everybody on the same page, generally what happens is you take fibroblasts, and they already have one or the other X chromosome inactivated. You reprogram that cell, and that's the iPS cells derived from that cell end up having one or the other X chromosome inactivated. What we found when we tried to reprogram protocadherin 19 cells is that we were losing all the cells that had inactivated the wild type allele and left the mutant one, so we were getting only wild type neurons. And so we've gone back, we actually tried several times and failed, so we went back and introduced a selection marker on so that we could get the kind of cells that we wanted. And we hope that will work, but we still don't know. with regards to what happens with X chromosome inactivation in iPS cells, as I think I may have told you before, the observation is that there is X chromosome, you get a line, it has a particular X chromosome that is inactivated, and it maintains that X chromosome inactivation over time, but after maybe 30 or 40 divisions, it starts losing that X chromosome inactivation. In fact, it seems to exist entirely. So it loses both inactivation of both of the X chromosomes. So, so it's something that you need to know about, and you need to do FISH or something else to actually make sure that the cell that you have still has the right X chromosome inactivated. So I guess that's, so it's, there's no question, it's far trickier to do it. I have to say though, we reprogram a lot of female cells, and just intrinsic reprogramming is easily done in females, but but if you're interested in the X-linked gene, then you might end up with skewing just because that mutation confers a disadvantage in cell culture.
Okay, thank you. Going forward on the downregulation of the GABA receptor, we were talking before with Carla and Professor Dalla Bernardina about the possible induction of high temperature of seizure. What about to induce to higher the temperature of
in vivo?
In vivo, yes.
In vivo.
Yes,
We
surely.
plan to do some behavior experiments and
Yeah.
to do that. But allora, noi stiamo ancora
Yes,
non abbiamo iniziato
we have not
esperimenti
yet
di
started
nel
experiments
senso che, al
of this
momento
sort.
abbiamo visto questo effetto
For the time being, we've just
sul
observed this
difetto
effect on the
nell'ippocampo del
defect in the
neurone.
hippocampus,
Adesso facciamo le
neurons in the
quindi
hippocampus.
vedere sia
Now we're going to look
l'attività
at the electrical
neuroni
activity of the
GABAergici,
neurons,
dei neuroni
both
elettrici.
glutamate
Una volta
and
fatto
GABA
questo,
neurons.
sul
Once
topo
we do that
andiamo a fare
on the
dei
rat,
test
we're going to run
specifici
tests,
sia
specific
per
tests
ritardo mentale
for
sia
mental
per epilepsia.
retardation and
E quindi
epilepsy.
in base
And
ai risultati
based
che
on the
otterremo
results that we
proveremo a fare
obtain,
un
we will
rescue,
try to
però
do a
non
rescue.
posso dire
But I
adesso
can't tell you
perché è
now.
chiaro
I
quindi
mean,
che il
it's
nostro
quite
obiettivo è
clear that our
quello,
goal
ecco,
is
però al
precisely that,
momento
but for the time being, we're quite far from it.
sì, certo, per forza, animale. Perché queste cose si possono fare solo quando si libera
I
l'animale.
think
C'era
can only
questa
be
cosa
done
del
on the
GABA,
animal
no? Che è
model.
la prima volta
Yes,
che lo sento.
I've heard this
Come?
GABA thing for the first time.
No, scherzo, che lo
No, I'm
sento in
joking.
pubblico.
I'm joking. It's funny to hear it in the
Lo
audience.
so, ci abbiamo provato,
We
riprovato,
tried many times, and we'll make it, I'm sure, at some point.
mi è venuto in mente
No,
invece per
but
quanto riguarda la terapia, la
as
possibilità di
regards
usare il
therapy,
vigabatrin.
what about
Il
the
vigabatrin è un
possibility of using
farmaco che usiamo
vigabatrin? This
molto
is a
negli
drug that we
spasmi,
use a
che ha
lot
un
in spasm
effetto
that has
GABAergico
a GABAergic
che
effect
diciamo aumenta
because it
il
increases
tono inibitorio,
the
quindi
inhibitory
facilita
tone,
la domanda più a
and so
disposizione.
GABA is more
Mi
available,
chiedevo,
etc. So I was
io non ho
wondering,
esperienza, non mi
I don't have
ricordo
any
nessuno delle
experience
nostre
with this,
pazienti
and I don't
trattate
remember
con
any of our
vigabatrin,
patients
non so
being
Nicola.
treated with
Alcune delle
vigabatrin. What about you,
nostre
Nicola? Oh, we have treated some of ours, yes, with that
Sabril.
no
drug.
effects.
No effect.
Sì.
Ci sono in sala
Yes.
almeno due mamme.
We have two moms here in the
Mi
audience.
pare con pochi effetti.
I
mi
think
pare,
that
no?
the effects were very few and far between.
Clara.
No, anch'io dicevo l'età era importante, sì, secondo me
Yes,
l'età di
age is
trattamento
important, age at treatment,
anche utilizzando gli stessi
using the
farmaci, ma
same
un'età
drugs
diversa,
too,
quindi
but at
può
a
avere un
different age can lead to a different
diverso.
effect, definitely.
Guarda, noi
Well, look,
diciamo che il modello che utilizzeremo adesso in vivo,
this
questo
is
di
the model that we're
vedere
going to be using
utero
in
come
vivo, the
allora,
in
rispetto al
uterus.
knockout che cos'è?
So
Abbiamo un modello di mosaicismo.
we have a mosaicism model. We have wild type cells and other cells.
Non ti so dire
So I
adesso
can't tell you
se
now
i nostri
whether
topi che hanno
our
questo
rats
difetto
that
neuronale
have this
nell'ippocampo,
neuronal
presenteranno un
defect
tipo
will
di epilepsia o
present
di ritardo
a
mentale.
phenotype
I
of
nostri gatti
mental
non
delay
l'abbiamo
or
fatto,
epilepsy.
però
We
lo
haven't done that
sapremo
work yet,
presto.
but we
E
will
quindi
soon find out.
momentaneamente non
So,
hanno crisi.
right, at the moment they have no
E
seizures,
quindi lo
right?
sapremo,
Right, yes. And
questi
that's
esperimenti li
why these are experiments that we're going to be conducting with a Genoa-based lab. They're epilepsy experts and experts in mental retardation, but I expect there to be a phenotype. I mean, an animal with such an evident defect is bound to have a phenotype. These are consolidated data. They're completely unbiased, totally unbiased. Okay, another question. Could we analyze the limbic system? Because if you look at the semiology of seizures, having analyzed seizures of at least 40 girls with a mutation of PCDH, what seemed most interesting was the very early involvement of the limbic system, the gyrus, and in the ECG that came up as well. So we thought that that ought to be one of the key points. Yes, you're right. I'm actually gathering information on that because very few people know how to do this in uterus technique in Europe. Very few people. There's a lab in the Netherlands that does this. And we want to look at the results on the hippocampus first, after which the idea is to selectively inhibit the expression of the PCDH. But, you know, if you act on the hippocampus, you pretty much interfere with the whole network. Yeah, the whole network. Based on what I hear, theoretically, there could be models made which involve the hippocampus without the involvement of the cortex, right? Yes, we could be selective about it. You can be selective about it and target specific areas. Okay. I guess if you put all clinical cases together, they may suggest that there are two different networks that could be responsible in the population. And it may very well be that those who have generalized chronic seizures or those that have cluster seizures and febrile seizures do best. And maybe they are different from those who have an involvement of the hippocampus alone. Although, of course, we need to consider the maturation process because evidently, when you modify things in the rat, I don't know whether you're going to be able to compare and extrapolate that data and apply it to man. Yes, of course. We're talking about an animal model, but it's clear. In the rat, we look at the development stage or process. So, of course, yes, I agree with you.
Yeah, I think that one of the things that we have to consider is that, in fact, you know, a knockout mouse does exist, and it's, as far as we can tell, it's completely normal. We had a lot of trouble generating anything, and now there are some issues with the mouse. It still expresses the message, but it doesn't have any of the protein left, and so. I actually was wondering, and I have to tell you, this reflects my skepticism of just sort of in utero electroporation and behavior, because when you do in utero electroporation, you only catch a small number of neurons, and you kill a bunch, and so it's great for like migration and things like that. But if you want to do behavior, I think you're probably better off investing in a conditional knockout where you can selectively knock it out in particular parts of the brain, because otherwise you're going to be facing a sort of uphill battle against what the sort of the field, right?
Yeah. Also we want to do, it's not easy, but we want to do also in acute. We want to knock down protocadine in acute with the stereotax injection, local injection, and we are doing the behavior study for the another gene, and we made the knockdown acutely for SHUM4, and we found an interesting phenotype. So we hope to see something also in adults, and in the two different way we are working now. But I agree with you that it's not easy, but it's not impossible.
I mean, let me just say that you know, mouse behavior and rat behavior is really, really noisy, right?
Yeah.
And electroporation is really, really noisy. And so if you introduce one noise and another noise and you put them together, you end up with really noisy assays. And I just tell you this because when we look at, you know, we want the most robust data possible, right? Because if you really want to develop a treatment, right, you need to have an assay that is really well characterized, because ultimately this is a $200 million project, right? And so I would say it's actually really worth spending. I know it's slower, I know it's more painful, and I know it's more expensive, but it is worth doing those kinds. I mean, the kind of clean genetic knockout in parallel, if the community can do it. I mean, it is definitely worth trying it, because that behavior is going to be cleaner, and it'll just be an easier model to then test drugs, to do other things, you know? Yeah.
Okay.
I agree with you. There is some problem about that, and in the lab we have different animal models, because we made an animal models for TSPAN7, and we published first, characterize the genes, and then now we are doing the animal model. But because first we have to think that it's not easy to make a good correlation between in vitro and in vivo results. Okay, this is the bottom line about all the results in the science. And maybe you have great results with iPS cells, with the neurons, culture neurons, but when you go in animal models, maybe you don't have a phenotype. Yeah. And this is the big problem, I know. And we spoke with Joseph about his animal model, and actually he didn't have any phenotype. And for this reason, we decided to do the two different approaches, because agree with you that is nice, but now with the electroporation, we will study development, and definitely maybe there is some effect, and we have to be careful about the results. And also, in parallel, we also will do with the stereotactic to see in acute. First, we analyze electrophysiology, and we will measure the current, the tonic, the inhibitor, and then we will do, agree perfectly, but, and this I agree, and because I have also experience about that, definitely, but.
Maria, can you also hypothesize to try drugs on young current in the when you're performing the electrophysiology?
Assolutamente, sì, però come si suol dire, non si può dire che si ha il gatto nel sacco se prima non si è fatto l'esperimento. Quindi prima vediamo un difetto, certo, e poi possiamo pensare.
In prospettiva futura,
in prospettiva,
certo. And one more, I would like to have your opinion, Professor Igorashi. What about your opinion? Yeah? Because I know that you are quite expert on IPS and...
Thank you very much. Sorry for my English is very poor. But I think in this disease, I guess the most important thing is some kind of modifying factor. That is, probably I think that probably determines the disease severity and phenotype. So more than 10% of our patients had some kind of mild abnormalities in MRI, brain MRI, that's suspecting a kind of migration disorder. So, but that is not significantly involved in seizure activities. So I will present in my presentation after this. So, but I guess a kind some very significant modifier will be involved in the severity. So we have to so focusing on such a modifying factor. Right.
Okay, thank you very much. Thank you. Ok, come si sono messi con i tempi? Ok. Perché poi magari dopo che parliamo della clinica verranno tutta un'altra serie di domande, di questioni sulla parte più clinica. Certo.
Carla, quando hai detto che
Carla, when you
il
said
trattamento
that in
farmacologico può
a
avere
drug treatment,
hai detto che un trattamento
now you
farmacologico
said
può
that the
avere un
drug
diverso
treatment
effetto
may
a
have a
seconda
different
del
effect
timing
depending on the
dell'età
timing
a cui viene
and the age
somministrato.
at which it's been
Intendi dire che
administered.
se lo dai più
Now, does this
precocemente
mean that
ha
if you administer it
un'efficacia in base alla tua
earlier
esperienza?
in life, this may
Non è
be having a different outcome?
Era un'ipotesi, diciamo, più basata sull'esperienza.
That was a hypothesis which was mostly based upon my clinical experience, because some drugs, whenever administered early, sometimes they don't work. And then if you reuse them later on in a different developmental moment and at a different age, may have a different efficacy and a different outcome. Much depends on the combination of the treatments that we can have. Sometimes it's association or combination treatments, other times it's monotherapies. So it's only a clinical suggestion. So if you administer them much too early, maybe you could try them again later on, in a later stage of life. Sometimes there are different systems, compensation systems that are being generated, so the drug tends to interact with this different compensation system. The GABA, for example, and this is well known, has an excitatory effect in the early days of life, rather than having an inhibitory effect. Later on, instead, it becomes inhibitory. So my comment was only having to do with the clinical aspect. Well, I was thinking of that compensation phenomenon that you were hinting at earlier on. It is quite possible that the seizures may be activating some cellular patterns through the genetic modulation. Yes, there is a so-called kindling phenomenon, kindling in epilepsy, which is very much visible in the animal model. So if there is a focus, a seizure focus, a second focus has been generated.
So I have a question, a clinical question for you. Do you find that there is a resistance to particular anti-epileptics over time? So do people sort of develop resistance and then you need to change their anti-epileptic and you have to do this repeatedly?
Yeah. I mean, that certainly happens with the benzodiazepines, especially with the clonazepam, nitrazepam, clobazam, all the benzodiazepines. There is over time, you need to do a washout period, and then you might really go back to the same benzodiazepine and obtain the same results. That's probably the best example I can identify for this habituation mechanism. And,
but in Dravet, that's very common. We also have a Dravet project, and there are many families where it's effectively intractable, right? They've gone through it, every single anti-epileptic, none of them really work, and But I was wondering if the same thing is true for protocoadherin 19 families or children.
Well, it's possible. I mean, in our experience, we couldn't really find a specific combination that was more effective rather than others, or a combination that really worsened the seizure frequency or the seizure duration, as it might happen for the Dravet syndrome with the Tegretol or phenytoin worsening the seizure frequency. That's not my experience. I couldn't really find any specific combination for either effective or not effective, and probably the drugs that really needs to be changed over time and do a washout period is probably only the benzodiazepine. In my experience,
as it might happen also in other
in other situations.
Other
Probably
situations.
the only benzodiazepine that might lasts for a longer period is the clobazam, frisium in Italy, that really the habituation or whatever mechanism is less
Less
evident.
severe.
It's less
I see.
evident. Even if there are some different scheduled in the treatment that I think we can discuss it later. Yeah. Okay.
So they don't develop like tolerance to valproate. They don't. No.
They don't.
Well, I was wondering if
Yeah.
if you know, protocoherine 19 mutation carriers develop
Some
sort of
specific habituation.
specific habituation to specific things.
Yeah.
Because,
don't
you know,
think so.
many people, as you probably both know, many people are well treated with existing anti-epileptics in terms of their seizures, at least, right?
Yeah.
But there is a subpopulation that effectively become insensitive to all of them. And so from a kind of drug development point of view, we're interested in, you know, unmet medical needs, right? So.
Yeah. What is my thought is that really we don't know at this stage if patients that are, as you told, insensitive to drugs would be insensitive. Probably it is the natural course of the disease, different. Patients with possibly drugs with good effects probably might only have a different natural course of the
Yeah.
disease. It's really early to say something about this.
What about the hypothesis to study children rather than mouse by some functional neuroimaging system? Because in some way this kind, almost in some cases, this epilepsy is suggesting the activation of some systems more or less sensible at different age and more or less sensible to some occasional but recurrent condition like fever and hormonal modification. So some studies like the study done in Ring 20, thinking that there is the possibility of GABA involvement in order to evaluate if there are some differences during this period of seizures in cluster and outside the period of seizure in cluster, because during the seizures it's difficult to do a magnetic
Sure.
functional magnetic resonance.
Yeah.
But between the recurrent seizures, when the child they are anyway frequently in status, not without motor manifestation, but perhaps it's possible to observe some kind of differences between the ictal and the true interictal period.
Yeah, so I you know, I don't have a huge amount of experience with this. I can tell you the results of one study that was conducted at Novartis around this. So I can't give you all the details, but basically the general conclusion was that there was a lot of variability across individuals with seizures caused by one specific mutation, and I would say that it wasn't. You know, the variability reflected the clinical presentation more than it reflected the mutation. So And so it wasn't obvious that there was something common, because most kids with epilepsy mutations end up with multiple classes of seizures. And so to some extent, the imaging reflected the class of seizure more than it reflected whether, so it didn't, I think we decided it didn't tell us that much about the mechanism because it was noisy, and also because we just couldn't look at enough people because it was expensive and long. But I mean, that was one example. It's not to say that you shouldn't do the experiment. You need to do the experiment because otherwise, who knows, right? It could well work, and it might be, revolutionary. We just don't know, I guess.
Yes, I was thinking about that. Carla, I was thinking about that, yes.
Study
Yeah.
using
A tracer of a specific,
tracer
yeah.
specific for the
Yeah.
GABA, as we do we did a similar study in Australia with the acetylcholine receptors.
Yeah, yeah.
And perhaps we could do a PET study, interictal PET study, of course, and maybe periictal PET study, and see if there is any special hypometabolism, maybe or hypermetabolism, who knows, and find out the structures, the anatomical structures of possible seizure onset.
But I think it's not so difficult to plan something, a PET scan, no? That was I was thinking about the neuroimaging hypothesis. Yes, yes. More than fMRI. I think it's best. The best way could be PET scan. Yeah. The ictal. Yeah.
But again, unless you do the ictospect at the beginning of the cluster of seizures, then you get a completely different pattern because the brain activates completely different at the beginning and at the end of the cluster. If at the end of the cluster you get seizure from the right temporal, at the end you can get frontal, left and right, and so there is a progressive activation and recruitment of different epilepsy foci, and so, but I mean, that's going to be difficult, yeah.
Right.